we'll get started with the material for today. Were there any particular problems with this last homework assignment? Okay, everyone found this one relatively easy? Okay, so I'll hand out the next homework at the end of class. But in the meantime, I guess we will get going with the material today, and this is kind of intended to wrap up the simulation part of the class by going through another one of the case study lectures where we look at a particular kind of system where people use many of the sorts of techniques we've been covering over the past few weeks. In particular, what I'm going to cover is a case study of the topic of simulating cellular biochemistry. So over the past few weeks, we've seen a fair number of different models. So we've seen some examples of discrete models. Can anyone tell me some discrete simulation models we've seen? Well, we've seen Markov models, so those come in a discrete time Markov model version, and also a continuous time Markov model, which we would also think of as a discrete model, even though it has continuous time, because there's a discrete state set. We've also seen discrete end simulation, which is a more general way of simulating some of these kinds of discrete systems. And we've seen various continuous models. And with our continuous models, what we've seen are basically versions of differential equations. So we saw ordinary differential equations, partial differential equations, which we would typically use in the simulation sense when we're trying to bring more independent variables, especially spatial variables, into the kinds of things we simulate with an ODE. And most recently, we saw stochastic differential equations, which are a technique we can use if we want a continuous model, but we also want to allow stochastic behavior, so randomness in that model. So these are some of the different kinds of methods we've seen, and actually most of these have been used in one way or another in studies of cellular biochemistry, so trying to build simulation models for studying how chemistry works in a cell. So what I wanted to do is go through some of the ways different models of these kinds have been used for this particular kind of system that I hope illustrates some of the values and trade-offs between these different methods, and also some of why I go through these kinds of methods in this class. So any questions about any of these methods before we get to that? Okay, well, let's start then by talking about a simple kind of system. Let's imagine that for the first pass at simulating a cell, what we're going to assume is that a cell is kind of a uniform bag of well-mixed fluid. So a cell is a fluid, it's got various chemicals mixed in it, and these chemicals undergo reactions. And we might imagine that we're interested in a particular kind of reaction system. So maybe what I'll say is that we're looking at a Michaelis-Menten enzyme system. So we'll assume we've got a substrate and an enzyme, and these form some intermediate species with reaction rates K1 and K2. And the intermediate, we can assume, can break back down, so it's possible for the enzyme to release the substrate without acting on it, or it can irreversibly chemically modify it to produce a charged state I star, which only we will assume has a forward rate K3, and that I star state would then represent the enzyme now holding the substrate after it's been modified in some way, and then finally the substrate would release, or the enzyme would release the substrate, which is now a product P, and then the enzyme is free to participate in other reactions, and we'll assume that has some rate K4. So this would be a basic kind of biochemical system that we might have going on in a cell that we might be interested in modeling. And we'll use this as an example of something that we might be interested in studying in a cellular model. So on the assumptions that I described to you, that we're, for the moment, pretending a cell is just a uniform, well-mixed bag of fluid, what would be a good kind of model for this? How might we represent that? how might we simulate it? Well, a good starting point would be to use an ODE model. So we might want to assume that if we're using this very simple model of a uniform well-mixed cell, then basically you can just think of there being some average concentration of S and E and I star and P in the cell, and we can describe the time evolution of the system in terms of these reactions based on these average concentrations. So we could come up with law of mass action equations. So we could say there's some equation that describes how the substrate evolves versus time. And can anyone tell me, based on these reactions, what that would look like? So what is ds dt for a mass action model? These are the reactions.
Minus K1 S.
Would be negative K1 times S plus E plus K2 times I?
Yeah, so you multiply the S and E, but basically we lose S through a forward reaction that has rate K1, and then that's multiplied by the concentrations of the reactant, so SE. And then we gain S through the reverse reaction, so I breaking down at rate K2 times the amount of I. So that would give us a mass action equation that would describe how S changes as a function of time. And we could go through and do the same thing with all of the other reactants. So we could say that there's an equation for E. So dE dt would evolve with E being lost at a forward rate minus K1 SE. It would be gained through this reaction, so plus K2I. And it would also be gained through this reaction when the enzyme releases the product. We now get the enzyme back to be reused again, so we also get a plus K4 I star. And likewise, we could say that di dt is K1 SE minus K2I minus K3I. Di star dt is K3I minus K4 I star, and finally dp dt is equal to K4 I star, because we only get P being produced by this reaction as a rate K4, and would be based on the concentration of I star. So in general, if we're trying to do a model like this, you would always put it together by looking for all the reactions that create a particular reactant and all the ones that consume the reactant, and then we would look at what's the rate of the reaction and what concentrations does it depend on. And this is pretty much all that you would have for any kind of mass action equation we're trying to develop. They pretty much all look something like this. So any questions about that? Okay, so that is basically the simplest kind of model you could use. And the earliest attempts to build whole cell simulators, the simulators of complex reaction systems in cells, pretty much did this. So you would give them the set of reactions you cared about and the rate constants and initial concentrations, and they would set up the mass action ordinary differential equations and then do a simulation of that system. So there were some basic tools, some of the earliest ones available for this, such as a tool called Picasi and another one called eCell. And these tools, in their first versions at least, did basically exactly this. You would specify the reactions you cared about, and they would model a cell by doing mass action equations for it. So, any questions about that? Okay, so that is kind of a first pass, but there are some pretty serious problems with the assumptions of this model. So it's unrealistic in some important ways. Can anyone tell me some of the ways that this model is unrealistic? How would a real cell differ from the assumptions of this model?
It's not uniformly distributed.
Yeah, so a real cell would be non-uniform. It would have a heterogeneous spatial environment in a few different ways. One of the simple ways you can implement that heterogeneity of the spatial environment is to take account of the fact that cells generally have compartments to them. Depending on whether you're looking at a eukaryotic or prokaryotic cell, maybe different sorts of compartment structure. But in a typical cell, we're going to have different regions of the cell, and reactants may not move freely between these regions. We might have very different concentrations of some of these molecules in some parts of a cell versus another. So the next kind of step that these models took was to move to what's called a compartmented ODE model, which is kind of a simple way to still keep ODEs as the basic machinery of the model, but get to this sort of compartmental structure. And we can kind of illustrate that by imagining if instead of having that uniform cell, we had a cell that had some kind of boxes in it. So there would be a nucleus box, and you could have an ER box, and a Golgi box, and a cytoplasm box, and so forth. And then what we would want to be able to do is to describe our reaction systems, accounting for the fact that different things might be happening in these different boxes, or at least that you might have different concentrations of different sets of reactants in different boxes. And that turns out to be something we can model pretty easily through some basic extensions of our ODE model. So the simplest thing we need to do to turn an ODE model into a compartmented ODE model is basically just pretend that each of our reactants is a different chemical in a sense, if it's in a different compartment. So we could say that substrate in the nucleus we'll call S sub N, and substrate in the ER we'll call S sub E, and we just pretend that these are two different chemicals. And then if we were interested in how these move between the nucleus and the ER, we could add reactions, like saying that there is a reaction that converts S sub E into S sub N with some reaction rate K. That would be a transport reaction, but from the purposes of our models, it's really no different than any other kind of chemical reaction. It would have some intrinsic rate, the actual rate of change versus time in these components would depend on that rate and the amount of thing in the starting compartment. And so we could pretty much take the model we already have and amend that to become a compartmental ODE model. So to see how that might work, let's take some basic assumptions about the enzymatic reaction system I was describing here. And let's imagine that what happens is the following. Let's imagine that the enzyme we care about is stuck in the Golgi. So that's always sitting in the Golgi. And the substrate, we'll imagine, is maybe produced in the ER, and then it has to transit into the Golgi for the enzyme to act on it. Once the enzyme acts on that, we get product, and then we'll imagine that product has to move to the cytoplasm to do whatever it's supposed to be doing. So that would be an example of a more sophisticated kind of system we might want to describe with a compartmental model. And that's something we can put into still our normal ODE language. So any questions about the model or what we're trying to do at this point? Okay, so let's see then what we would have to do to come up with this model. So one of the first things we're going to want to do is amend our reactions here to describe what we're talking about in this model. And the way I've described it here, most of it's relatively simple because I've described the model in such a way that basically all the interesting stuff is happening in the Golgi compartment. So what we would pretty much want to do is take our initial reactions, S plus E goes to I, with rates K1 and K2, and then just turn that into the reaction S sub Golgi plus E sub Golgi goes to I sub Golgi at those rates. And we could say the same thing for I goes with rate K3 to I star. We just say I Golgi goes to I star Golgi with rate K3. And I star Golgi goes with rate K4 to P Golgi plus E Golgi. And we've now described what it means for these reactions to occur in the Golgi, and then we just need to create a few additional reactions to describe the other parts of the system. So how would we describe the movement of substrate from the ER to the Golgi? what would that reaction look like?
SE with some K.
Yeah.
K. Or SG, yeah.
Yeah, so let's just call that K5. So we've now got a reaction K5. And we could do the same sort of thing at the other end with the product. We could say PG goes with rate K6 to PC. If we wanted this to really work more realistically, we might need to add in a couple of additional reactions. So we might want to assume that there is a synthesis reaction. One way we could write that is to say that nothing goes to SE with some rate K7. And we could also, let's say, propose that the product, maybe it gets secreted from the cell eventually, so PC goes with rate K8 to nothing in the auxiliary system. What we now have are a set of reactions that describe our compartmented model. So does this make sense to everyone? All right, so at this point, what we have is something that we can pretty much convert into our ODE language, just like we did before. So we could say, more or less, it's going to look like the reactions we already had. So for example, we could say DSG dt is going to be minus K1 SG EG plus K2 IG. But then we have to add some additional reactions there. And in particular, what we're going to need to assume is that if we've got this reaction creating SG from SE, we're going to need to add one more term to this equation. So what would that term look like? Well, we would need to add something that describes a reaction with rate K5. So we're going to have a K5 here, and it's going to depend on the concentration of S in the ER. So we'll get a plus K5 SE term. We've now got a description for the rate of change of SG as a function of time. And we could do the same thing with the EG dt, and it's going to basically be the same as it was before. We'll get the minus K1 SG EG plus K2 IG plus K4 I star G. The I and I star also pretty much work like before, so we get the IG dt is equal to K1 SG EG plus K2 IG, see that's not right, minus K2 IG plus K3, or minus K3 IG, all right, and we've got a di star G dt. This one also, we're basically just adding G's to everything, so K3 IG minus K4 I star G. And then we've got the product, which we have to change a little bit. So dPG dt is going to be equal to K4 I star G. And what do we have to add to that? Have to account for this reaction. So we've got a minus K6 PG, so that's taking out the rate at which PG turns into PC. And then we just have a couple of additional things we have to add to this. So we've got all our equations for describing what's happening in the Golgi, but we've added effectively a couple of new variables. So we need a DSE dt and DSE dt. We're going to have some rate at which SE is produced. Can anyone figure out what that term would look like? Nothing becomes SE at rate K7. With a rate of K5 because it becomes SG, so minus K5 SE. And then the last thing we need is a dPC PT equation. Can anyone tell me what that would look like? This is a K6 and a K8.
K6 times PG,
have a K6 PG, which is the rate at which it's produced.
And then minus P8 PC.
Yes, minus K8 PC. And that is going to give us then a complete set of ODEs that now describes our compartmented model. So does this make sense to everyone? All right, so at this point, we've basically got a model just like before we put in the compartments that we can simulate with our standard sorts of ODE algorithms. So what might be an algorithm we would apply to either of these models? What's an appropriate algorithm for an ODE? We simulate ODEs with any of our basic numerical integration methods. Depends on what properties we want for our model. But for a system like this, usually we're going to have a stable system. So maybe we would want a backward Euler method, so it has nice stability properties. Or maybe we want higher accuracy, so you go with a Runge-Kutta method. But basically, numerical integration, or standard numerical integration techniques would do the job for any of these. Right, so this compartmented kind of direction is the way that some of these sorts of systems went. So I mentioned the Capazzi system, and in fact, Capazzi in a later release went in this direction. So it added in a way of doing compartments within the model, and then you get to kind of a next level of realism in the model. All right, so that is kind of a next step of evolution of these systems, but they're still kind of unrealistic. So in what way is this model still unrealistic? What are some of the problems with it? We can actually go a little further with the issue of inhomogeneities in the spatial system, because even within a compartment, you're going to have some inhomogeneity across space. So maybe you have a cytoplasm, but if product is being released from the Golgi, it's just diffusing. You might have a very high concentration near the Golgi, a low concentration elsewhere, or maybe it's being transported somewhere in particular. Maybe it has to go to the surface of the cell, and it's actually ending up there, or whatever. But you can have these kinds of discontinuities, or not discontinuities, but inhomogeneities in space for our reactants. So we would often want to go to another level of complexity to capture that sort of behavior. So how might we do that? How could we change our model?
Add subcompartments.
What's that?
Add subcompartments.
Yeah, you could break the compartments down to a finer and finer level, but often what you would really want to do is take that to kind of the limit of just saying that we have continuous space. Now, often the way you would really represent the continuous space computationally is to discretize it, so it becomes equivalent having lots of little compartments. But at least in terms of how we would describe it mathematically, we would pretend at least that we were talking about continuous space, and that we might have concentration of product, let's say, potentially different at every point in here as a function of spatial coordinate. So what kind of mathematical model would describe that sort of system? So that's where we would be bringing in typically partial differential equations. So we would want to now assume that each of our molecules has some reaction rate, or has some concentration that's a function of the position and the time. So we would have potentially P in the cytoplasm at spatial point xy or xyz and time t. And then we need to start describing time evolution of a system like this, and that's where we've seen partial differential equation models coming in as our main simulation technique. So suppose we wanted to take exactly the system I've described here, but we'll just throw in the assumption that the reactants are diffusing within this space. So maybe we'll say that we have reactions that are transporting things from let's say the PG to PC reaction, we'll say it transports this only from the spatial point right on this side of the membrane to the spatial point on the other side, and then otherwise we'll just let things freely diffuse. How would we amend our differential equation models to capture that? What would you write differently to say that P is diffusing within the cytoplasm? Well, we can start with the model we have there. So we can say dP sub C dt. And since this is a partial differential equation model, I'll make this partial of P sub C with respect to T. We're going to say that that is equal to what we have here. So K6 P sub G minus K8 P sub C. And then we'll want to throw in something else to say that this is diffusing, so that this, as a function of x, y, z, and t, is somehow diffusing. So how do you describe diffusion in a differential equation system? When we looked at models of pure chemical diffusion, what did the partial differential equations look like for that? Well, the standard equation that we'd seen, remember, was if you're describing diffusion of a chemical within some space, we would say dC dt is equal to, let's say, some diffusion constant delta times the Laplacian of C. And does anyone remember what a Laplacian is?
With the second derivative, or partial derivative?
Yes, it's the sum of all the second partial derivatives with respect to the spatial coordinates. So that would be second partial of C with respect to x squared plus second partial of C with respect to y squared plus second partial of C with respect to z squared. So if you throw that into this model here, so we come up with, let's call it delta sub P, the diffusion constant of P, and then we could throw in this extra term, second partial of P sub C with respect to x squared, second partial P sub C with respect to y squared, and second partial P sub C with respect to z squared. Then we get something that now describes a spatial system in which we have reactions producing and consuming these, as well as diffusion of the P sub C within that space. So does this make sense to everyone? Now, in terms of actually implementing it, things would get a little more complicated because we do have to worry about things like that the transport reactions are only acting at certain spatial points, and then the diffusion reactions acting at other points. So in terms of the kinds of techniques we've discussed, you'd sort of have to think of this in terms of having boundary conditions at each of the boundaries between compartments, and then maybe a separate set of differential equations within each compartment. But basically you can throw it all together and pretty much use any of the kinds of methods we've already seen to simulate these sorts of systems. So let's say, for the sake of argument, we were trying to take this equation and simulate it with a scheme that was first order in space, or let's say second order in space and first order in time. What might that look like? What might the basic iterator equation for this look like? Well, we would want to start with something that looks like, let's say, the amount of P sub C at time point n plus 1 and spatial point xi, yj, zk is going to be something we would compute from, let's say, P sub C at time step n and spatial point xi, yj, zk. And then we would want to say that that is plus, let's say, delta t, if we were using a forward Euler method, times an approximation to the derivative at that point at step t. And what would be the approximation we would put in for the reaction part of this? And this is basically a reaction diffusion equation. put in one part that has this component, which is basically saying that the amount of new PC we produce at that spatial point is going to be delta t times the rate at which this is gained through this PG to PC reaction. This obviously we would need to remove if we weren't at the boundary, but if we were at a boundary where this reaction was occurring, we could say that this would be xi yj ck. So we could say this is being produced by some rate that depends on the amount at that boundary. And if we weren't at a boundary, we need to remove that. We would say that if we're at the other appropriate boundary, we could say that this is removed at some rate Ka, Pc, and xi yj ck. So it's removed at a rate that depends on the current amount. And then what would this term look like? How would you approximate the second partial derivative of P sub c at this spatial point? Well, we'd have our scaling coefficient, so delta sub p, so that would be our diffusion constant. And then we would have to use a numerical derivative approximation of the second derivative of P sub c with respect to x. And the way we've seen to do those approximations of the second derivative are that we would have P sub c at time point n and spatial point xi plus 1, yj zk, plus P sub z at time point n and spatial point xi minus 1, yj zk, minus 2 times Pc at point n, time point n and spatial point xi yj zk, all divided by some delta x squared. And that would give us a numerical approximation to the second spatial derivative of P sub c with respect to x. And then we would put in analogous terms for the y and z. So we'd have basically the same thing but with y and the same thing but with z. And putting all of that together, we get a basic iterator equation for describing a reaction diffusion version of this system. Does this make sense to everyone? Okay, so this also is something that people working in this field of whole cell modeling took as kind of the next step in the evolution of the field. So the Capacity system I was telling you about actually went this direction as well. They came up with something called the model extender for Capacity, or MEG, which was basically a way of taking a Capacity model and breaking it down into a really fine set of compartments, so effectively doing a PDE model through very small compartments. And there was a more recent simulator called the virtual cell, and the virtual cell simulator started directly with this kind of spatial diffusion model, and they had been doing this specifically so they could come up with very realistic structural models of a cell. So you can really do realistic sizes and shapes of all the components of the cell and get a nice model for how that behaves. So that was kind of a next step in the evolution of these sorts of systems. All right, so that is a more realistic model. It's getting much closer to what would actually happen in a cell, but it's still kind of unrealistic in some ways. So can anyone tell me in what ways this might still be an unrealistic model? One of the ways that this is unrealistic comes from the fact that cells are pretty small. And because a cell is small, you would often have very few copies of particular kinds of molecules. So all of these mass action things basically depend on the fact that if you've got very large numbers of molecules, all the randomness in which reactions occur when kind of averages out, and you can pretend you've got a continuous deterministic system, but really that's not what's going on. You have individual molecules going through individual reactions, and so the system ends up really kind of noisy. And when you're looking at the scale of single cells, that noise in the system is actually kind of important. Some kinds of reactions won't occur or won't occur in your model if you don't assume the reaction systems are noisy. So a classic example of this is, if any of you have studied molecular biology, you might be familiar with the phage lambda lysis lysogeny system. It's how a bacterium infected with a virus basically decides that it's time to start producing virus and blow up the cell and all of that. If you don't have stochastic behavior in the system, you can't model what's going on there because it's basically a kind of random switch that has to get flipped at some point. And what flips it is basically just random noise. You happen to get just enough of the right reactions happening that something gets pushed over a particular level, and then the cell switches from lysis to lysogeny and starts producing virus and blows up. So that's been kind of a big trend in studies of cellular biochemistry over the past maybe 10 to 15 years, is understanding these kinds of stochastic reaction systems. And so if we want a realistic simulation of what's happening in cellular biochemistry, we want a way of capturing that. So what are our options for simulating stochastic systems? How might we improve on this approach? Well, we've basically seen a couple of ways of doing this. One of them would be to take the kind of continuous models we already have and add stochastic behavior into the continuous model. And if we're doing differential equations, what we'll end up with is a stochastic differential equation model. So if we were trying to come up with a stochastic differential equation implementation of a system like this, then one of the things we could do would be to say that any reaction we're interested in, so such as the K6-PG reaction, instead of saying that we have dPC dt equals K6-PG minus K8-PC, and let's just ignore the space again for a moment, what we would say is that this kind of captures the average rate at which PC is produced and consumed, but then we have some random fluctuation on top of that. And a typical way you might describe these is if you look at a small-scale model of the chemistry, we can say that reactions are more or less exponential timed events, which makes numbers of reactions versus time the Poisson process, and that ends up implying that the standard deviation in the number of reactions we should have in any given unit of time should be about the square root of the total number of reactions. And what all of that is getting to is that we could throw in a stochastic term for each of these that would be of the form something scaled by K6-PG, or square root of K6-PG. Yeah, you can just leave that. And then what we're going to have is some noise term, let's call that dW1. So that would give us an amount of noise that would be in magnitude about the square root of the rate of production of these reactants. And then we could have another noise process that describes noise in this reaction, so minus square root of K8, PC, dW2. And if this were a noisy reaction system, we would want to account for how that noise shows up in other reactions. So if we were producing PC, we would also want to account for in the dPG dt reaction where we have a comparable term minus K6-PG. We would then have to add in a comparable term minus K6, the square root of K6-PG dW1 to reflect that whatever extra reactions we have here producing more PC are reflected in extra reactions. How we would take this kind of ordinary differential equation version of the model and turn it into a stochastic differential equation that would now capture noise in the reaction at an appropriate level to describe the stochastic behavior if you have, let's say, just a few copies of the PG molecule. So if you have maybe 100 PGs per cell, which might be a realistic number for a typical kind of protein, then that would give you a more realistic model of what chemistry would look like in that cell and how it would vary cell to cell. So does that make sense to everyone? Any questions about this?
That's similar to the random ones we did on Tuesday, correct?
Yes, this is the same kind of technique. So with a stochastic differential equation model, we would have these noise terms, and then if we were trying to implement something like that, then what we would do is say, well, really, I suppose I should rewrite this to be strictly correct, to say that it's dpc equals this dt plus the noise terms, and dpg equals this dt plus noise terms. But then if you wanted to simulate that, we would use the kinds of techniques we saw in the last class. So we would come up with an iterator that would look something like P sub c at step n, or n plus 1 is P sub c at step n, plus a forward Euler term for the deterministic parts, so delta t k6, pg at step n, minus k8, pc at step n. And then each of the noise terms, we would have to have a random operator, random variable describing that. And those would always have the form of square root of delta t times a normal 0, 1 random variable. And I'll just call that z1 here. And then we would also have the scaling factor here, square root of k6 pg. And then this would be another independent normal, so minus square root of delta t z2, square root of k8 pc. And the reason I'm calling this z1 and this z2 is that you would have to keep track of the fact that this z1 is the same one you would get in this other equation because it's the same noise process. So we want to ensure conservation, but this one is an independent sample. It's a different noise process. Does that answer your question? Okay, any other questions at this point? All right, so this is one way we could go to describe noisy chemistry, so the kind of noise you would get when you start getting to small numbers of reactants. So almost all of the techniques we've seen for these kinds of simulations have actually shown up in some cell simulator or another. This is the one exception. As far as I know, no one's actually implemented a cell simulator that uses stochastic differential equations. There's no reason someone couldn't do that, so I mentioned this as one option available to us. I keep hoping one of these years, one of you guys will pick that as your final project, because I think it would be a neat thing to do, but it would actually be a relatively difficult project and not necessarily recommending it. But that's one option if we want to handle stochastic systems. But it's still a little unrealistic in some cases, because if you start getting to really small numbers of molecules, this is going to do some things that really couldn't happen. So this will occasionally tell you that you might have half a molecule of P, and of course you can't have half a molecule. You've either got 0 or 1 or 2 or whatever. So sometimes if we're getting to really small numbers of molecules, we want to use a different kind of model. Can anyone think of how we might do that? What kind of model have we seen that would be a good way of describing a system where we have just a few copies of each molecule in the system? Well, what I'm proposing there is really that we're moving to a system where it matters that the state set is discrete. So if we're keeping track of a certain number of molecules, or let's say we're keeping track of these molecules S and D and P and so forth, and each of these has a copy number that's from 0 to 10, let's say, then there are 1,000 possible states, so it becomes a discrete state set. And that's something where we would typically use a Markov model to represent it. We're no longer worrying about a continuous state set. But for this kind of chemical system, if we want realistic chemical kinetics, so realistic changes in the system versus time, we're generally going to want to use a continuous time Markov model system. So that's kind of our other major alternative. And I want to spend some time talking about how you would implement a system like this as a continuous time Markov model system. All right, so to see how that would work, let's imagine we have what I'll call a generic reaction. So we've got a set of reactants, x1, x2, up through xn. I'll just ignore that specific example of the enzyme for the moment. Then a generic reaction we could describe as saying that there are A1 x1s and A2 x2s, etc., up through AN xn's, and these are consumed to produce B1 x1s and B2 x2s, etc., up through Bn xn's. So that would be a generic kind of arbitrary reaction system. And we can assume that we're looking at a model where each transition in the model corresponds to one of these generic reactions. So we have a set of possible reactions that can occur, and then transitions or state changes in the model would correspond to a reaction. And for a system like this, a very obvious model of the state set would be counts of our reactants. So we'd have n1 x1s, n2 x2s, etc., up through n sub little n xn's. And then what we would want to do is come up with a Markov model system where whatever reactions we have describe how you can transition between states in this kind of model. And the basic state transition is not really that complicated. We would say if you have this generic reaction that we're trying to implement, then a transition is possible if you have at least basically, if you have n1 is at least a1, and n2 is at least a2, and nn is at least an, and so forth. And if you have that, then the transition would be of the form n2 up through n sub n, and that would transform your state from n1 to n1 minus the number of x1s you consume, which is a1, plus the number of x1s you produce, which is b1, n2 minus a2 plus b2, etc., up through n sub little n minus a sub little n plus b sub little n. So this would be one transition in your model. And then for any starting state that has enough molecules to produce the reaction, and any reaction, you get one possible transition in your Markov model. Does that make sense to everyone? All right, so if we're trying to implement a continuous time Markov model, we need to know more than just the transition. We need to know the rate of that transition. And we can also assume that there's some reaction rate k here, and you can derive the Markov model rate, so the rate of the exponential random variable that describes waiting time for this reaction, based on knowing the reaction rate for a standard kinetic model and knowing the concentrations of each of your reactants. So in particular, if we're trying to produce this reaction, we're going to assume it has a waiting time described by some exponential rate lambda. So the waiting time is an exponential random variable with rate lambda. And that we would assume is going to be proportional to k. So that would kind of make sense. If you want this really to work out, you don't quite use your standard k, because k is usually going to be specified in something like molarity of the different molecules, or moles per liter, or something like that. To get the right scaling constants, you basically need to represent k in molecules per whatever volume you're simulating, but that's just some scaling constants. And then we need to multiply this by all the possible ways of implementing the reaction. So if we have n1 copies of x1 and the reaction needs a1 of these, how many ways are there to pick the x1 reactants for this reaction? Going to be n1 choose a1. So we need a1 copies of x1. We had n1 available, so n1 choose a1 is the number of ways of picking those. And the reaction is going to have a rate. So So the rate until the next one of these is going to be proportional to the number of ways of picking those reactants, and it'll be the same with all of the others. So we multiply by n2, choose A2, and so forth out to n sub little n, choose A sub little n, and that's going to give us a scaling factor for the rate. So the more copies of our reactants we have, the faster the reaction is going to go. Does that make sense to everyone? Now, there's one little issue here which comes up because what you need to make these small-scale Markov models work out doesn't quite agree with the standard convention for how people represent the reaction rates in large-scale chemistry. So normally if we have a mass action model and you have a reaction like A plus A goes to B with rate K, then the mass action model will have a term that looks like dA dt equals minus K A squared. So we assume that the rate constant is defined so that the appropriate amount of A is A squared. But if you're looking at a small scale reaction, what we would have is effectively the concentration of A choose 2. And the point I'm trying to get at here is that this thing is sort of equivalent to A squared over 2 rather than A squared. So it's just kind of a convention that you end up off by a factor of basically, well, what it ends up being really is the factorial of A. So if you want this to work out correctly, you have to add in this kind of extra scaling factor, A1 factorial, A2 factorial, A3 factorial, AN factorial. But basically what I'm trying to illustrate here is that if you know the rate constant for your large-scale mass action models, or your standard sort of kinetic rate constant, you can figure out this lambda. It's just a one-to-one mapping, and the math is not really that complicated to do. All right, so basically what we end up with then is we've got our state set, we've got our transitions, we know the waiting time for our transitions, so we've got everything we need to specify a Markov model, continuous time Markov model for this system. And if you run that model, what you're going to end up with is something that, in the limit of large numbers of reactants, should behave exactly like the ODE model, but for small numbers of reactants will give you a much more realistic representation of randomness and discretization in the reaction. Does that make sense to everyone? All right, so that's kind of another direction we can go with these kinds of models. But if we go this way, you need to get into some issues we've so far largely ignored when talking about continuous time Markov models, and that is that these things can get complicated enough that you need to worry a lot more about the algorithms you use to simulate your models. So I want to say a little bit about the algorithms we would use here. So in chemical systems, these are often referred to as SSA models, which refers to something called the stochastic simulation algorithm, which is a standard way of implementing them, or they're also known as Gillespie models, because Gillespie was the person who invented this for chemical systems. And when Gillespie first proposed this, he proposed a couple of different possible algorithms you can use for this. So one of them is what he called the direct method. And the direct method is something that should be familiar to us for if you think back to when we looked at how you would implement a continuous time Markov model. Basically what it says is you identify all of your transitions, so we'll call them edges E1 through EK in your graph. You calculate the overall rate of transition out of your current state, which is going to be some lambda, which is simply the sum over the lambdas of all of the edges out of your current state. So remember that if you have a continuous time Markov model and you have rates of transition lambda 1, lambda 2, lambda 3, and so forth, the total rate of transition out of that state is the sum of the rates of all of the possible transitions. That's all this is saying. We sample waiting time, so let's say tau is exponential with rate lambda. And then we pick which of our transitions we want to use from the discrete distribution proportional to the rates of the possible transitions. So lambda 1 over lambda, lambda 2 over lambda, and so forth. That gives us our probability of taking transition 1, transition 2, and so on. And then we just update the state according to whatever reaction we picked, and then we go back to the initial state. So this is one of the ways we had described previously for implementing a generic continuous time Markov model, and it works fine with a system like this. But it can be inefficient in some cases. So if we're looking at a complicated chemical system where maybe we've got lots of reactants and lots of possible reactions, we don't necessarily want to run through all of these different possibilities when we are trying to figure out what happens next in the system. So Gillespie had proposed an alternative that sometimes works better that he called the first reaction method. And the first reaction method sort of looks similar. We start by identifying the transitions. So we look at our current state, figure out which of the reactions have the right set of reactants that they might be the next thing that occurs. And then for each of these, what we do is sample a waiting time. Ti gets exponential with rate lambda sub I, the rate of the corresponding reaction. We pick the minimum, update the state to reflect that the corresponding reaction has occurred, and then go back to step 1. So this is a slightly different way of implementing the system, but it has some of the same efficiency problems. On each step of the system, we have to run through all of the possible reactions that might be happening next. And it's a different calculation. In this case, we take all of the exponentials and pick the minimum time one, as opposed to figuring out the overall rate, so the distribution of the minimum of all of them and picking that once. But it has some of the same efficiency problems if you have a very complicated reaction system. So those were the two kind of original methods for doing this, but subsequent researchers proposed a method that will work better when we have systems where lots of reactions might be possible at any given time. So can anyone think of how that might work? How might you improve on the efficiency of those methods? Well, suppose, for the sake of argument, we're looking at a system that has 1,000 different reactants, but no reaction involves more than maybe 3 of them. What is something inefficient about this we can improve on, or inefficient about either of these? Well, we can take advantage of the fact that exponential random variables are memoryless to say that if we've got lots of things that might happen, and we sample times for some of these things, and they don't happen on the next steps, they're not the next thing that happens, then as long as the next thing didn't change this distribution, it's actually fine to just sort of leave that alone and let that time sit until it becomes the next thing that happens. And what I'm getting at here is that you can implement these kinds of things more efficiently by turning it into a kind of discrete event model. And in particular, what we can do is just once figure out all of the possible reactions, set up an event queue that has all of those reactions waiting to occur, and then each time a reaction occurs, we only have to pick new times for a few things whose times were affected by that most recent reaction. So in other words, what we end up with is something that's called the next reaction method. And the next reaction method works as follows. We just once identify all possible transitions from our starting state. So let's call those E1 through Ek.